we're going to take a look at specific immunity, and in particular, we're going to be talking about the T lymphocytes. Now we know that there are different T lymphocytes that have different responsibilities, and so we're going to start by looking at APCs, antigen-presenting cells. And we'll start with the antigen-presenting cells we're going to do in blue. And remember, we talked about them having an MHC2 site where they display the epitope. And so then our helper T cells, we're going to make those pink, just so that we can keep them separate. So this would be then our first antigen-presenting cell. We'll call it number 1. And then what's going to happen is we'll have this helper T that I've drawn here will recognize. Remember, they're very, very specifically recognize antigens, and so it won't just be any helper T. This particular one can recognize that displayed antigen on this antigen-presenting cell, and once is not enough for a cell to be able to, for a helper T to be able to do anything with that. And so really what has to happen is they have got to then recognize a second PC, number 2, all right? And again, there will be a piece of that antigen displayed in an MHC2 site. And there will also be what we call a co-stimulating protein. And so this time, when this same helper T comes to a different APC and recognizes the MHC complex, it's also going to interact with this protein, this that I just drew, this protein right here, and we're going to call that then co-stimulation. Co-stimulation, and again, that is when the helper T that we start with up here moves down and actually finds another APC that has the same antigen displayed, and it will recognize that second one. Okay, now we're in business, because once a helper T is co-stimulated, what's going to happen is it is then considered, we will activate that, and that will give us then multiple, this is the selective cloning process that we talked about. So multiple helper T cells, T helpers, these are all T helpers. And it's also going to give us a number of, as we talked about in class, T memory cells. Okay, now the cells that are actually going to be going out and doing the work this time around in this particular situation, this battle, will be these helper Ts. Now, we know they're not going to do the killing, but wait until you see what they do. Okay, so remember the T memory cells, they're just going to hang out and wait for something in the future to happen. All right, so we've got these activated helper Ts then, and what's going to happen is they're going to then go out, and there's going to be all these APCs out here that are displaying that same antigen. Lots and lots of APCs. Again, they've all got the same antigen they're displaying. Well, so this is like going to be number 3, number 4, up until the millions, okay? Well, these activated helper Ts are going to come over here, and What do? Well, they recognize that antigen. They recognize the antigen. These are the activated helper T's. All right. So as they recognize the antigen then, what's going to happen is they're going to secrete what we call interleukin. Now this falls in the category of cytokines. Cytokine called interleukin. All right. And we listed all the different things that will cause to happen. We're going to recruit a whole lot of the non-specific players, the natural killer cells, macrophages, neutrophils. A lot of people are going to get drawn to the site as a result of the interleukin. Now, I'm also going to show you how this interleukin affects the cytotoxic T cells and B lymphocytes.